base stuff though?
I'm taking guys.
Sorry, I'm 10 minutes late.
No, you're fine. Perfect.
Thank you. No, my kids came back from a music tour on Sunday, and I sent a couple of stinging emails to my youngest at the state of his room. Took pictures, and got off the bus and was just down in the dumps. And then I started feeling bad for making him feel bad the whole trip back from San Diego. So I said, was it my emails? And he said, no. Well, his room is really tiny. It's the smallest room in our house. It's about 10 by 10. And so it's got room for a bed, and it has a huge aquarium and a wardrobe for his clothes in the closet. And so there's not room. So everything there is actually better than his sister's room because everything was everywhere. But by the time I took his clothes out and sorted them, they covered the entire floor of my bedroom, which is 3 times larger. And so I'm tired of doing your laundry kind of thing. So I got off the bus and said, no, Mom, it wasn't your text. He said, I left my leather jacket in the restaurant, and I left my new headphones, $150 headphones that he'd helped buy in the hotel.
Yeah, it would be mad.
So he gets down on himself really easily. And so he called the restaurant, and they checked the bathrooms in where he'd been sitting, and it wasn't there. I said, it was a DoubleTree hotel. So that's a nice hotel. I said, just call Lost and Found. And since Sunday, his best friend had borrowed it overnight and placed it on the desk with his breakfast ticket on top. Well, he had got his ticket a different place, so he didn't see it. So it was kind of a mix-up of communications. It wasn't really himself forgetting it that badly. So he's been calling since Sunday, and Lost and Found will never answer. He keeps getting forwarded to them. So I was trying to help out and putting my husband on it. All right, so we're looking at the structure of the stomach, and typically the stomach is always modeled as what we see it as in the cadaver, although our stomach and our cadaver is a little bit different, but it has this kind of J shape. All right, if you were to look at x-rays of stomach contents, it doesn't look like this at all. It's long and more of a tube shape, kind of a wide dilated tube, and it can be half again as long as this. But we typically draw it looking like this. So we have the esophagus coming in just a short section below the diaphragm. There's actually an opening in the diaphragm for the esophagus, known as the esophageal hiatus. We'll come back to that. Well, we were talking about the esophagus. So, ever heard of a hiatal hernia? We were talking about inguinal hernias. That was down in the inguinal ring. And just got an email from Dr. One that one of our cadavers students spotted what might be an inguinal hernia. So asking for a consult on our cadaver. Not that we'll do surgery or anything, but. So when there's a weakness in the muscle area, when a baby cries a lot, it can develop an umbilical hernia after birth. So an esophageal hernia is a weakness in the central tendon of the diaphragm such that abdominal contents can not necessarily enter the thoracic cavity, but at least push up into it if they don't actually enter it. One of the most interesting surgeries I read about was the development about, it's been now 15, 12 years in San Francisco, where babies were being born with congenital esophageal hernias, hiatal hernias. The problem with that is they're developing, and they actually end up with the organs up the stomach and parts of the intestines up in the thoracic cavity at the time that the lungs are developing. And so instead of developing the alveoli, the lungs develop as solid tissues. So then when the baby's born, they don't have the gas exchange surface areas, and it's a little hard to do lung transplants on newborns. And so doctor in San Francisco developed a process. Surgeries have been done in utero. We talked about the transfusions, where they would just put blood in the abdominal cavity or put a shunt into the baby's ventricular space while it was still in utero. In this case, the extensive surgery required better access to the fetus, and so the mother was essentially given an early C-section. and they put under general anesthesia. The uterus opened from top to bottom. This was done at about 4 months. No, so it's not too far along, but still extensive. And the amniotic sac opened up, and the fetus taken out of the sac, and its chest opened, and a Velcro-like patch placed to repair the hole in the diaphragm. The abdominal organs brought back into the abdominal cavity. This Velcro-like patch put in place. The baby placed back in the amniotic cavity, sterile saline added, the amniotic membrane, and all the layers of the sac, and then the uterine wall stitched back together. And the baby was given a 50% chance of survival. They would have had none at all if the surgery hadn't been done. And of course they had to do then a C-section because the uterus isn't going to repair itself in time to go through labor at that time, and then that was successful. I just saw an image of a surgery, a similar type of surgery, but done for a different reason. And I'll see if I can find it. It was posted to my Facebook. So during the lab break, the fetus was brought out at 26 weeks. No, 24 weeks maybe, somewhere around 24, 26 weeks. And surgery was done. And as the surgeon was just ready to move the fetus back, the fetus was actually still in the uterus. So the picture is of the surgeon's huge hands, the uterine wall, the uterus, and then this tiny little, the baby reached its fetus, whatever you want to call it, reached its hand up and grasped the surgeon's finger. And it's just this adorable picture of just that image right there. But it's not this surgery, so obviously they developed this technique to work for additional surgeries. Yes?
Is that hernia the diaphragmatic hernia that you're talking about?
Yeah, same thing.
Oh, okay.
But a hiatal,
With
yeah.
diaphragmatic hernia.
Yeah, it can occur in adults, but this is a congenital, in the infant, was congenital.
Yeah, because we have like babies that we deliver
Uh-huh.
that have them, but we deliver them early
So that they can
we
then do a
have-
repair.
yeah.
In this case, it was up so high that there wasn't any functional lung tissue development that would have occurred. So obviously with the Velcro patch, they can go in there as the infant grows and increase the fix. So that where this is obviously on the left side, we have a rise, pouch-like structure that develops in the stomach. And then the rest of the stomach comes down to form the body and narrows to form this region. So this area, there is remember we talked about the sphincter, so our first sphincter was voluntary with the spicularis oris. There is a slight thickening, it's not very noticeable, of the circular smooth muscle at the joining union between the esophagus and the stomach, known as the cardiac sphincter. It's also common to see it called the gastroesophageal, if you like to write words. Or you can't remember, it's the union between the stomach and the esophagus. So maybe you don't like all those letters, but you can't remember the term cardiac, so either term suffices. The cardiac region of the stomach is kind of a ring around this cardiac sphincter. Then the fundus, remember the uterus had a fundus? That top part, the bladder had a fundus. Well, the stomach has a fundus as well. And when you look at an x-ray, let's say someone is having gastroesophageal problems, you look at an x-ray, that space usually is not filled with food or with the barium milkshake, and is usually black because it contains air. It's not very common to have stomach contents up in that region. This is known as the body. And then the tapered region is the pylorus. And now we do have a very extensive thickening of the inner smooth, inner circular layer. As we join the stomach to the first part of the small intestine. And this is known as the pyloric. So the purpose of the pyloric sphincter is to, the cardiac sphincter is going to open as soon as we do peristalsis and food moves down there. So the purpose is more to keep the acid contents of the stomach from, moving up into the esophagus when you cough or stand on your head or whatever. Because it's going to open at any time as food comes through. The pyloric sphincter is under control of nervous system and hormones, as is the activity of the stomach muscle, because we only want a small amount of stomach content, known as chyme, to move into the duodenum at a time. The stomach content is very acidic. The enzymes that do most of our digestive breakdown, our chemical breakdown, require alkaline pH. So if we dumped a cup of this acid into the small intestine, it would take a long time to neutralize that acid pH. So we allow just 1 or 2 tablespoons at a time to come in, and that's the role of the pylorus, is to regulate that amount that comes in. If we look at the lining of the stomach, you'll notice that it's thrown into these longitudinal folds of mucosa, and we use the same term that we use in the bladder and in the vagina, rugae. Okay. Now we do have a plasticized stomach. I'm not very proud of it. It looks kind of weird. Stomach. Is it haggis? The Scottish that they stuff the stomach haggis with oatmeal and meat, other stuff. Anyway, just made me think about eating this thing. This is the rugae, okay, the longitudinal folds. I was unbiased. I didn't make a face when I said it. All right. So now let's look at that plan, the GPAT, so that you missed all that on Zoe, didn't realize. So what we call the organization of the layers of the wall of the digestive tract, they all have 4 components: mucosa, a submucosa, a muscularis externa, and a serosa. So we're going to be looking at this pattern for each of the organs and components of the digestive tract. Now, I this stomach is something that particularly, I don't know, I'm going to put my name to this one. Now remember, in the stomach is where we change our epithelia from non-cornified stratified squamous to simple columnar. So on your study worksheets, study guides, and on the review slides, there are light microscope slides that show this transition. It's called the esophageal gastric or gastroesophageal junction. There's an immediate sharp change from non-cornified stratified squamous to simple columnar. Okay? Where else would you see this change? Where else do we change from between stratified squamous and simple columnar epithelia?
Your trachea?
Not to simple columnar. We would go to respiratory, to the pseudostratified. Nope. That goes from transitional to stratified squamous. I'm thinking within, I'll make it easy, I'm thinking within the digestive tract. No? So if I put a slide up there and you saw non-cornified stratified squamous on one end, and then an abrupt change to simple columnar on the other, it could be one of 2 places. The rectum, exactly. So rectum to anal canal. So the anal canal is non-cornified stratified squamous, and the rectum is simple columnar. Okay? So you need to be able to tell which one you're looking at. They're both simple columnar. However, a couple of clues. When we look at the stomach, the pattern of the mucosa is such that we have gastric pits and then gastric glands. Now I'm drawing it rather schematic. It's not going to be this pure and simple. That's the surface. All right, so it's like you're walking around in holes, big holes in the ground. And then of course you would have your muscularis mucosa. That's the mucosa of the stomach, or the gastric mucosa. On Tuesday, when we compare it to the small intestine, Our surface area also has holes, our intestinal glands, but then it's thrown up into villi. Remember the model I showed on Tuesday of the mucosal plan? So we have these villi, muscularis mucosa. So this is the plan of the small intestine. Still simple columnar epithelium. Okay? The plan for the rectum is similar to the small intestine but without the villi, similar to the stomach but without the gastric pits. So it's just glands. With the mucosa. Colon and rectum. You'll see diagrams similar to that on your lecture exam, and I'll ask you which organ you're looking at. Okay? But keep that in mind, we're just looking at this portion. So for the stomach, here are the gastric pits, and here are the gastric glands. Now, remember I said that although we have simple columnar cells in the stomach, we don't have goblet cells. The gastric pits are lined by mucus cells, and I'm going to come back and do this histology right now. We're just looking at telling the difference between the two. So we do have mucus cells, but they're not goblet cells. The rectum is going to have goblet cells, about 25%. So many it actually looks like about 75% of the cells in the rectum are goblet cells. So those are the characteristics that you want to look for as you try to figure out, are you looking at the junction between the stomach and the esophagus, or are you looking at the junction between the rectum and the anal canal? You want to look at the characteristics. The non-quantified stratified squamous is going to look exactly the same. You're going to want to look at the characteristics of the simple columnar epithelium. All right. So starting with the mucosa, we're back to just looking at details of the stomach now. So we have these gastric pits and gastric glands. Simple columnar cells is our epithelium. In the gastric pit area, these are identified as mucus cells. All right. Down here, they're mucus neck cells. I'm not going to test you on the neck cells, but you'll sometimes see that they're still mucus cells. Okay. And then we have in the gastric gland, this is down within more of the lamina propria, we have almost triangular shaped cells. Into my red, that are parietal cells. And these are the cells that secrete hydrochloric acid. So hydrochloric acid, of course, is what contributes to the acidity of the stomach contents. The only thing I can think of for why we need the stomach, unique among all the other organs of the digestive tract, to be acidic is to kill off bacteria and viruses in our food, to provide a chemical defense. Because it really is disruptive to the digestive process. Yes, the acid will activate our major protein digestive enzyme in the stomach. It changes pepsin into pepsin. We'll see next week. But But very little of that digestive activity goes on in the stomach. We rely more on our pancreatic protein enzymes than we do the stomach one. So the salivary amylase is activated in our mouth, provided you chew more than a minute. Requires a pH slightly acidic, about 6.8 to 7. That is completely denatured when it hits the stomach. The acid content of the stomach totally denatures that enzyme, so it no longer works. So our food is in our stomach, becoming acidic. Yeah, a little bit of gastric lipase and pepsin will work, and then it goes to the small intestine. And none of those enzymes in the small intestine work at a pH of 1 or 2. They would be denatured. They work at a pH of 8 to 9. So we have to dump a ton of bicarbs from our pancreas to bring the pH back up again. That's a big switch in pH. So we can actually do chemical hydrolysis, digestive hydrolysis in the small intestine. And it's like, why did we do all this in the stomach just to turn around and negate it all? And so the only thing I can think of is it's kind of this vat of acid to take care of pathogens. The digestive enzymes are coming from most of all the other cells, which are chief cells. And we'll come back again to this on Thursday. Down typically closer to the base of our gastric gland, we have endocrine cells. You know, the stomach, like the kidney, is not typically thought of as an endocrine organ, but it is. It's mixed. And so these are identified as entero, which really refers to intestinal, but all of the endocrine cells of the digestive tract are referred to as enteroendocrine cells. And more specifically, because these cells release a hormone known as gastrin, they're also called G cells. So those are these bright yellow ones shown here. Now we have loose, irregular connective tissue that's present surrounding all of this. Our lamina propria. And in contrast to the small intestine, the gastric glands tend to be packed very close together. I've not drawn it that way, but there's usually very little space between them, so you're not going to see much of that lamina propria if you compare a stomach slide to a small intestine slide. Right? And then finally we finish off the mucosa with our muscularis mucosa. And this helps to form those rugae. That would be this structure right here. All right, so we got some new models, thanks to Jeff. He likes anatomy so much. So these blocks back here, there's a couple of different sets. Are of the various changes. So here you can see we're switching from stratified squamous to simple columnar. All right. So you have to figure out, is this anal canal to rectum, or is it esophagus to stomach? Then we have these structures here. So this is, anybody I was going to say what a hazard, I'm not going to put water in if you make the wrong choice, so it's really not a hazard. Anybody want to guess what you're looking at here based on what I drew on the wall? Pardon?
The colon?
But the what organ?
Is that the colon ring?
No. So these are villi, and these are glands here.
Small intestine.
So that would be small intestine. Okay. This would be the muscularis mucosa here, and submucosa. And so this would be stomach with the mucus, the gastric pits. Notice on the surface is more similar. It's bumpy with holes, but you don't have the distinctive villi structure. So these would be like the little shrubs or bushes coming above line. So we have those. We have two sets of that. And then we have another one here where they're in sequence. So if you figure out where in one place, you can just go above or below, proximal or distal to figure where else you're at. So this kind of shows the layering of the GPAT, the mucosa, submucosa, muscularis externa, and serosa. And then on the outside here, it shows a little bit more detail of specific cells or structures. So these are models you can start looking at for the tissue components. Okay, our submucosa, I'll just put submucosa up here before I erase and move to muscularis externa. Submucosa is our standard. Okay, there's nothing particularly unique about the submucosa of the stomach. So it's just going to be a dense, irregular connective tissue. So that's more similar to what the models are looking at like. So parietal cells, this actually should be up here because those are not mucus cells. When we look at the histology of the stomach, and I'm not going to get this detailed, but just to explain some of the things that you will see, we can tell where we are in the stomach, whether we're in the fundus or the body or the pylorus histology-wise, because of the difference in the length and depth of the gastric gland pits. So this is a gastric pit here. This is a gastric pit. Because these gastric pits being so long, this is down towards the pylorus, which is of course closer to the small intestine, this becomes very difficult for novices looking at the histology of this to tell the difference between these gastric pits versus intestinal villi. Although intestinal villi would have goblet cells and these do not. So if I show you a histology slide and expect you to identify it as the stomach, I would show you one that's more similar to this, where you can see the parietal cells and the gastric pits are not as easily confused with intestinal villi. Okay, because we don't have tons and tons of microscope time to spend hours and figure out and find differences between these. But I do want you to recognize that there are variations that occur within the stomach. All right, let's look at the muscularis externa. So notice we have an extra layer here. Okay. I'll leave this diagram up for a little bit and erase, well, take a picture if you want. I need to erase some of it to diagram the muscularis externa. I'll erase the right side of the board, but take whatever you need. So instead of our plan, our model, which has 2 layers of smooth muscle, because of the churning and twisting and wringing role of the stomach and mechanical digestion, we have 3. So we start out with an innermost, closest to the submucosa, an innermost oblique layer. So we look at the stomach. This layer is going to be going in this direction, kind of at a diagonal across the stomach. Then we have an inner circular